Hello, good morning, good afternoon, everybody, and welcome to our very first ImmuneWatch webinar. Before we get started, a quick note on questions. Feel free to ask questions during the webinar using the Q&A feature, which you can find by clicking in the bottom right on the activities button, a triangle, square, and a circle, and then click on Q&A. Feel free to ask them during the webinar, but we will address them after the presentation. Yes, I have the honor to introduce Pieter. Pieter Meysman is a professor at the University of Antwerp and has been working with T-cell receptors for almost 10 years now. With TCR, his lab also pioneered one of the very first TCR epitope prediction algorithms. He's also a co-founder and part-time CTO at ImmuneWatch, and today he will share some insights on how he has been applying TCR epitope predictions in his work. I'm excited to have him here today, and the floor is yours, Professor Meysman.
Thank you for that introduction, Sander. So, before we start, I'm going to be talking a lot about TCR epitope prediction algorithms. Now, this might be a concept that many of you are really familiar with. So just to kind of get an idea of, let's say, the scope of the audience, I'd just quickly like to do a poll on if people here have already used these kinds of algorithms in their own research or their own work. So normally you should be able to see in the Google interface a poll that popped up that you can just simply vote yes or no. But for those of you who aren't familiar, I'm just quickly going to briefly explain the concept. So really what these predictions, or at least the type of algorithms that I'm going to be talking about today, are really those that try to predict the epitope specificity of a T-cell receptor. Now, in most cases, what we're doing is we're actually training a machine learning algorithm that can then identify if indeed this, for instance, this TCR here that you see, can bind a specific epitope that we're interested in. For instance, this specific hepatitis B surface antigen. And then we simply want to have, is this binding yes or no? So this is a very typical machine learning problem that we can think of in this sense, where we have a set of T-cell receptors that do bind the epitope. So these are the ones in red over here. And they are separated by what is called a decision boundary from those that are in blue, so those that do not bind the epitope. So a quick look to see at the poll right now. So it's about 50-50, half and half. So some of you have already used these kind of algorithms, the other half have not yet. So I'm hoping to kind of share with you today how these different algorithms can be used currently, and how we have been using them to really uncover new and exciting things about the human immune system. So one of the things that if you've ever seen a talk by me before, that I talk a lot about is this difference between seen and unseen epitope prediction. I'm not going to spend too much time here, but just know that the difference is really in what the algorithms are able to annotate. So for which epitopes we actually can get an answer. And so most tools that are currently available fall into this seen category, where they can find known patterns for an epitope and find any TCRs associated with that specific epitope. So that actually means that the algorithms are actually limited to those epitopes that are already in the database or in the training data set for which they can make predictions. However, there's quite a lot of work being done on what we call the unseen epitope prediction, where we're trying to identify in general patterns, so for any epitope. So this would actually mean that this is an unrestricted algorithm that can actually predict for any combination of any TCR with any epitope, if there is binding, yes or no. Now, there's quite a few approaches these days to do this sort of predictions. And I'm actually quite pleased. I was also part of this organizing committee that actually generated this blinded benchmark for these algorithms last year, where they ran a competition trying out 53 different algorithms to see how well they do against a set of blind data. So this is very similar to these kind of other scientific challenges where you have a competition between different scientific labs to see what actually the status of current predictions. Now, they did recently publish a paper describing this challenge and the results. I do urge you to check it out if you want more details. But the main conclusion that I kind of want to point out here is that let's say the best performing algorithms were really good at doing this kind of seen epitope prediction. So it actually meant, so these are actually these little in black epitopes here at the bottom. For these, the top performing algorithms really performed quite well. However, for the unseen epitope predictions, so essentially for epitopes that weren't in the training data set, which were in red here, the top performing algorithms didn't perform so well, so this remains at least an open challenge as far as this benchmark is concerned. So when we're going to be talking about applications today, I'm really going to be talking about the applications of these seen epitope prediction models. And the one that I'm going to be talking about the most is ImmuneWatch Detect, which of course was developed by ImmuneWatch, which also scored quite well in this benchmark. Now, in general, so what we're trying to do is really linking TCRs to the epitopes. And the way that they typically would be used is if you have a T-cell receptor repertoire, so I mentioned you've just done TCR sequencing, you can then use them as that data as input to actually go and annotate those TCRs that you identified against specific epitopes that are in the database. So essentially you get this list of TCRs annotated with an epitope. Now, in practice, this looks like this. Imagine this is your output, your typical AIR formatted clonotype file where you have your different TCRs, their clone count and their clone fractions. And what you typically add then is an extra column that says, okay, this is the putative target epitope, as well as a score, which would typically be the confidence score of how certain am I that this TCR can bind this epitope. And this is something that you can already do today with the current, let's say, generation of TCR epitope prediction algorithms. But of course, this is simply just applying these methods. But what I want to talk about today is really how these tools can actually be used to really drive this kind of new insights into your TCR data. Because it is one thing to actually apply these algorithms, it's a different thing to actually use them. And I've kind of outlined 4 use cases here that I'm going to talk into a bit more in depth, being repertoire annotation, repertoire certification, response certification, and diagnostics. And just to have a quick opportunity for an additional poll here, I'd quickly like to survey, okay, what, let's say, is the main interest? I haven't really gone into too much detail here. So repertoire annotation is really where you get a TCR repertoire, and then you just simply have a look at, okay, what TCRs are present, can already identify which TCRs are against which epitopes. The second one, this response stratification, is really when you're looking at, let's say, typical longitudinal data, where you have TCRs, TCR data for a known therapy, and you simply want to do kind of immunomonitoring to see, okay, how good was my T-cell response based on these TCR sequences that I've now identified. The third option is this repertoire stratification, where we're really looking at, okay, what is distinguishing between different groups at the TCR level. And I'm going to talk a bit about how you can actually use these kind of TCR epitope prediction models to compare TCR repertoires between different individuals. And finally, let's say the last point is really on the diagnostics, where I can also use this kind of TCR epitope prediction models to do really diagnostics, to really distinguish between 2 cohorts with, let's say, a different disease. Now, it's okay if these concepts aren't too clear for you yet. I have plenty of examples in this presentation. So don't be shy to, let's say, take one now and change your mind later on. So just quickly looking at the poll, so we've got about half of you have already voted. Most of you seem to be interested in repertoire annotation, but it does seem to be widespread across the different applications. So I'm going to focus on everything a bit here, and feel free to ask me specific questions at the end, as Sander has indicated. So let's start with repertoire annotation. So this is, let's say, the most straightforward application that you can consider. And I'm going to talk a bit about a recent publication that we did, where we actually built a single-cell T-cell atlas, where we actually collected TCR data from 12 different single-cell studies that actually comprised 149 different samples. And this final atlas contained about 500,000 cells, so 500,000 T cells, where they actually both had matched gene expression data as well as TCR receptor data for each of these cells. So we did quite a lot of analysis on this, but one of the ones that I wanted to point out, which was quite interesting, was that we actually used this atlas because it was collected from so many samples to have a look at public TCR clusters. So what I mean here by clusters is really motifs within the TCR repertoires that seem to occur more often than you would expect across these different individuals. So this is distinct from public clonotypes, where we are really looking at a motif that is occurring across different individuals. And we actually did find several motifs that seem to be quite interesting and that occurred in a wide range of individuals. And one notable cluster that you identify, I'm showing it here. So this is the CDR3. I believe this was an alpha chain, where we actually found this motif, or at least instances of this motif, across 99 of our 149 samples. Now, given, of course, that these people all had different HLA types, or we assume that at least, we already know that it's going to be impossible that this motif is HLA specific. So we were kind of wondering, okay, if this isn't HLA specific, what exactly is going on here? Because we actually find more than 1,000 sequences across these different samples. Now, if we ran this, or at least these 1,000 TCRs through ImmuneWatch Detect, we actually saw that they all matched a riboflavin antigen, presented not on HLA, but on MR1. Now, for those of you who are familiar, of course, with T-cell biology, you already realize that what we actually found here, or what this cluster is, are indeed the mucosal-associated semi-invariant T cells. So this actually makes sense that we picked this up in our T-cell atlas because mate cells are typically everywhere. And of course, it is well known that they have a semi-invariant T-cell receptor, so therefore they will be a very prominent motif in our data. And we're actually able to confirm this prediction actually then by looking at the gene expression of these cells, because of course, since this was single cell data, we had pair data between the T-cell receptor and the gene expression. And we were actually able to confirm that indeed we found several markers typical for mate cells expressed in these cells that were predicted to be mate cells based simply on the TCR sequence. So I really like this example quite a lot because it really shows you that what we're getting as a prediction is really validated by, let's say, the other data levels that we can typically look at. So this was, let's say, the start. So this is what we can already do with repertoire annotation. Let's say the main application that we've been doing a lot of work on, or at least how we even got started with TCR sequencing in the first place, was on response stratification. Now, I've got a few cases here, but in general, what we're always trying to do is really track TCRs longitudinally. Now, TCRs, this is actually a unique aspect that we can exploit because a TCR is unique to a T-cell clonotype. This actually means that if we pick up a TCR in multiple time points, we can assume that they are derived from the same clonotype. And since the space of potential TCRs is so wide, we can really use this for longitudinal tracking. Now, what we're going to try to do is see, okay, how does the TCR repertoire change in response to a certain event? Typically, this will be a certain treatment. Very typically, this will be an immunotherapy like a vaccine. Because if we then overlay the epitope information, we should be able to see which TCRs are responding and how they relate to the therapy that we have induced. So if you look at this from a repertoire perspective, what we will typically see is that we have a repertoire pre-intervention where we have, for instance, we can annotate those TCRs that are associated with our immunotherapy. So for instance, those TCRs that we know respond to our vaccine. We then have our intervention, and afterwards we have our second time point, a new repertoire, where we should see expansion of T cells reacting to our therapy. So again, we should be able to see a difference in the annotations that we get from our TCR repertoires after this intervention. And the case that I'm going to discuss here, because this is a nice example, this is actually one of the first ones we ever did, is on the hepatitis B vaccine. So this is a well-known vaccine that is given quite often, and it is well known that there is a issue of non-responders with known risk factors like age, male gender, as well as HLA genotypes. And what we wanted to do was to investigate the TCR repertoire of people that got this vaccine to see if you could find any additional signals that kind of underpinned this response type, as well as to evaluate if TCR sequencing can even be used to evaluate the T-cell immunity response. So what we did is we actually recruited 34 individuals with no prior hepatitis B vaccination or disease history. We confirmed this using antibody titers to see that indeed none of these individuals had an antibody titer against hepatitis B prior to starting the study. We then gave them the vaccine, and what we then saw was that we could actually divide our cohort into 3 groups. We had our non-responders, which is what we kind of expected. And then we could also distinguish a group of late responders and a group of early responders. And the difference is here that, of course, hepatitis B is given in several doses, where the early responders were those that had a strong antibody titer even after just a single dose of the vaccine, while the late responders only had a somewhat lower antibody titer almost 6 months later. So we actually collected samples at different time points. We actually did quite a lot of detailed study on this, but what I'm going to talk about mostly here today is actually only this kind of TCR repertoire sequencing that we did on these individuals of their CD4 memory repertoires. Now, this here is, for instance, one of the outputs that we got from one of the individuals of this CD4 memory TCR repertoire. This is, of course, the first, let's say, 20 or 30 lines of a file that is almost 50,000 lines long. So we have plenty of TCRs, but as we know, these are all unique for each individual. So we kind of need to tease out, okay, which are indeed these vaccine-specific TCRs that we are picking up. Now, because, of course, this is not a good way of looking at it, the way that I typically regard this is by looking at it, let's say, from a network perspective. Because what you can do is you can actually encode every unique TCR as a single dot or node in this network. So this would be this gray node that you see here. So this is a unique TCR that we detected in our memory repertoire. And what you can do is you can actually make a link between 2 TCRs if they are sufficiently similar. Now, how you define sufficiently similar is, let's say, an interesting question that I'm not going to go into too much detail, but in this case, it's so simply having a single amino acid difference in the CDR3 region. So what you actually get is this very nice overview plot of a single repertoire from a single individual. And you can already kind of appreciate the complexity of the TCR repertoire if you look at this image, because it is a very dense repertoire. So we have a lot of different TCRs, if you consider that every dot is a unique TCR. And as well, you see also these kind of clusters popping up, which is something that is quite common if you're looking at TCR repertoires. Now, what we can then do is we can then use these kind of epitope prediction algorithms, because we had data for the hepatitis B antigens, to do predictions of, okay, which of these TCRs, which of these tens of thousands of TCRs are actually reactive against our vaccine. And what we found was, in this case, 10 TCRs that were reactive to our vaccine. So this really kind of shows you what kind of a needle in a haystack problem identification of, let's say, or annotation of TCRs typically is. So this is an individual that had gotten this vaccine, and we find 10 TCRs that are putative to be reactive against this vaccine. So in this case, it's kind of highlighted in red. I'm not sure if you can even see it, but you have, let's say, 3 red dots over here. We've got a couple of them up here, and we've got 2 of them down here. So this kind of already illustrates, let's say, the complexity of this problem, why you really need a good performed algorithm to even attempt this, because the moment that you're getting too many false positives, where you're actually starting to predict TCRs that aren't reactive to your vaccine, they will very quickly dominate the signal. But luckily for us, this actually did work out quite well. And in fact, if we then indeed have a look at, let's say, the before and after, if we simply count the amount of hits that we get before and after vaccine, we very clearly see that the number of vaccine-specific TCRs have expanded in this cohort. as you can kind of see here. So in this case here, we have T0, so this is our before state, and we have our T60, which is after 2 months. So this is 1 month after the second dose. And we see that for most patients, we see this kind of expansion, which we would expect if they had gotten a vaccine. And this is purely based on these epitope-specific predictions that we're going to be talking about today. So this kind of already nicely validates that even though this is really a needle in a haystack problem, we can pick up a signal of a T-cell response in these individuals. However, one thing that was quite surprising to us for this study at least, was that what we saw was that there were quite a few individuals that actually had TCRs before getting the vaccine. Now, remember, these were all naive individuals. We confirmed it with antibody titers that these individuals did not have any antibodies against hepatitis B, nor any prior disease history against hepatitis B. And more interestingly, if we then had a look at the differences between these responder classes that we had, so remember we had fast responders and slow responders, we saw that it was mostly these fast responders, so the people that actually had a strong antibody titer after there's a single vaccination dose, that had these pre-existing vaccine-specific T cells. And it actually gave us quite a strong classification, so actually good ROC curve, as you can see here, so a trade-off between sensitivity and specificity, where we could actually see that we got a good prediction distinguishing those fast responders from the slow responders, simply based on if they had these predicted vaccine-specific TCRs before getting the vaccination. So we did quite a lot of work on this, and we actually did quite a few follow-up studies here, which really delved a bit deeper into, okay, what is really driving the vaccine response against hepatitis B. I'm not going to go too much into detail here, because this would take us too much off track with the TCR sequencing things. Okay, so this is simply evaluating if can we pick up a TCR-specific signal based on a vaccine. However, this isn't typically the research question that we want to ask, because if we have a vaccine, we can already typically know if they are a responder, yes or no, based on a number of other characteristics. So if we're going to be using TCR epitope prediction models, we can actually make use of several other aspects to really delve deeper into what is actually happening in these specific individuals. And now one of the typical research questions that you may have is a vaccine, any kind of protein vaccine is of course comprised of multiple epitopes. And we can actually have a look at what epitope is actually inducing this response. And to really delve deeper into this specific research question, I actually have a different example. So this is actually a study that we did with the Centrum for Human Genetics in Tübingen, where they actually had a cancer vaccine they had administered to a colorectal cancer patient. Now, this patient actually already had, at the time of diagnosis, liver metastasis, and was actually already undergoing 2 years of chemotherapy, at which point the state had actually degraded, where more metastasis had already occurred, going even into lung and lymph nodes. So therefore, he was kind of selected as a prime candidate to be given in a new experimental peptide-based vaccine to really see what it would do and if it would actually help him. Now, what actually happened is they actually gave him this vaccine, which was fully customized to his specific tumors. So they actually selected out those epitopes that were specific to his tumor based on genomic sequencing. And after a year of treatment, he was actually declared cancer-free, so where there was no remission anymore of the cancer. So this was actually a good case for really delving deeper into, okay, what actually worked here, what was actually driving this response, because of course this cancer vaccine was made up out of different epitopes. So as I already mentioned, this was a personal vaccine that was cancer-specific for this specific individual, which was based on mutations that were observed in his specific tumor. And really the research question that we tried to address together with the researchers here was, okay, we have these epitopes that we integrated into our cancer vaccine. Which epitope was eventually responsible for driving this kind of response? This was actually quite a challenging question to answer, but luckily we could use the kind of the TCR sequencing to kind of come to the rescue. Because we had the typical setup as before, so they actually took samples before and after administering the vaccine, and actually did TCR sequencing both on blood samples as well as tumor tissue samples. They also actually took a subset of these samples to actually do epitope stimulation against these different epitopes, so that we actually had a small set of TCR epitope pairs to start with, to actually start doing these predictions. Because remember, we are actually working in the seen epitope prediction models here, so that actually meant we actually needed some starting data to actually get a good performance for our prediction models. Now, when we actually then applied these prediction models to these pre- and post-vaccine data sets, so I'm just showing you here the dynamics of 4 of these epitopes. So in these 4 different colors is each of these a dynamic of a single epitope. And what we see is, so we actually start with the pre-samples, where we've actually normalized everything to a value of 0. So to assume that we had no reaction to start with. And looking at the different dynamics following vaccination, we see a clear upregulation or a clear expansion of epitope-specific TCRs targeting peptide 19 and peptide 21. So this was clear in both the blood sample as well as in the tumor samples, where we see a clear expansion of TCRs targeting these 2 specific epitopes. And now what was interesting in this case was that they actually did follow up in vitro experiments to actually validate that indeed these were the 2 epitopes that were driving the immune response within the specific cancer vaccine. So this was actually quite exciting for us, because this was a direct confirmation that, first of all, our epitope prediction models seem to be capturing this kind of expansion, and that this was actually able to be validated in the lab when actually doing more typical in vitro experiments based on our own in silico predictions. Now, we can actually take this one step further. And so this is actually going to be my last example for response stratification, where we can actually delve deeper into the question of, okay, why do actually some patients respond differently to a therapy? Because if we are able to actually characterize the T-cell response that actually comes out of a therapy, we can actually make use of that to maybe dig deeper and to actually see, okay, where do we actually see a difference between those that have a different outcome? And so this is actually an example of a study that we actually did in my lab that was published just a few months ago, where we actually took data from this study that had already been done on AML patients, where they had gotten hematopoietic stem cell transplantations. And several of these patients had relapsed, while several went into clinical remission. And now one of the questions that we had, which was really driving all of these studies, was, okay, why it was a relapse and why was a remission in others? And we're kind of hoping that, let's say, the TCR repertoire would give us some answers to this question. But of course, as you know, every TCR is unique, so that actually made comparing these individuals quite challenging. However, we know that if you then do these kind of epitope annotations, we can then look at, okay, do we actually see any differences in the epitope annotations coming out from these individuals, given, of course, that we actually also had, again, longitudinal data. So in this case, because, of course, this was simply a stem cell treatment, we actually had to look wider at the different cancer antigens. So for those of you who are working on oncology, you should be aware that, of course, there's several antigens that are primarily associated with cancer. These are also let's say, the basis of many of these novel cancer vaccines that are currently coming out. And for our lab, or at least the lab that we're also currently working with, we actually had quite a bit of interest in WT1, so the Willem's tumor-associated antigen 1, which is known for being a quite, let's say, prevalent antigen in many tumors. So what we actually wanted to do was to actually have a closer look at, let's say, T cells that were targeting the WT1 antigen in the specific data set and see if we could indeed identify any differences between these different responder types. Now, of course, one of the big issues that we had was that, again, we are working with scene epitope prediction models, so it actually meant that we needed some sort of data. Now, currently, let's say in the public databases, there was very few information already available. So that actually meant that we actually had to do, let's say, experiments ourselves to derive WT1 specific TCRs. And since we didn't want to, let's say, sacrifice any precious cancer samples to actually do this, because while we did have cohorts available, we actually decided to actually set up a protocol to actually derive WT1 specific T cells from healthy individuals. So we actually did this together with the lab of hematology at the University of Antwerp and the University Hospital of Antwerp, where we actually stimulated the PBMCs of healthy individuals with WT1 specific epitopes. We then used actually tetramers to actually sort out those T cells that were epitope specific, followed by TCR sequencing. And through this manner, we actually set up our own little database of WT1 specific TCRs that we can then use to actually start doing this kind of scene epitope predictions on any kind of cancer dataset, including the one that I described at the start. And so in this case, what we actually used, so this was actually TCR, so kind of the precursor of ImmunoDetect, where we took the repertoires from these different cancer patients, as well as healthy donors, and we simply predicted, okay, how many WT1 specific TCRs did we identify in each of these individuals. And what we actually found was quite interesting. It's going to be quite detailed. I'm kind of hoping that you can kind of see this clearly on these slides. Otherwise, I do suggest that you have a closer look at this, the paper. I have the citation at the bottom. Because what we observed was, in this case, this is again kind of this like network representation of common clusters that occur across these different individuals. And what we've kind of overlaid here in the color are different WT1 specific epitope annotations. So in this case, we had in blue and in yellow WT1 epitope specific TCRs. And what we actually found was that there were, let's say, about 7 very prominent clusters. So these are clusters, so TCRs that are very similar to each other across different individuals, that we could assign at least one of them to be WT1 specific. So you can see them here, they're kind of with these dotted lines. So these are across different individuals, we find common clusters that you could assign as being WT1 specific. And now what we then saw, if we actually compare these different responder classes, was that as expected in the healthy individuals, so these are these 2 first columns here, we find very few of these WT1 specific TCRs and very few hits of these TCRs in these specific clusters. What we found in the, let's say, in the relapse were more hits as expected, because of course these were people that actually had AML, so we actually do expect some WT1 specific TCRs. However, they were concentrated in just a few of these clusters. We see here for this individual, we see that this person had a lot of WT1 specific TCRs, but they were, let's say, congregated in a single cluster. However, if we then actually have a look at the, let's say, the remission patients, so these were these 3 individuals, what we actually saw was that there were actually TCRs across these different individuals, across these different clusters. So that actually meant that they actually had a much broader response against a lot of different, let's say, variants of the same TCRs, targeting the same epitope, but having, let's say, different binding motifs or different recognition motifs as predicted by these epitope-specific algorithms. So this seemed to indicate that what is really driving, let's say, remission is really having a large diversity of TCRs that can recognize the same epitopes. And that these kind of relapse patients might have been hindered by the fact that they only had a very specific or limited amount of TCRs that could react against these epitopes. However, of course, further studies should be able to indicate this, and this is something that we're, of course, continuing now in our lab with, let's say, larger cohorts as well. Okay, So this was, let's say, response stratification, where we are looking at a specific treatment and how these people have changed, or how these people's TCR repertoires have changed in response to that. In the next example, I'm going to talk a bit more about repertoire stratification. So in this case, what we have are different cohorts of individuals, where, for instance, we have, let's say in the typical case, we have a healthy group, we have a diseased group, and we want to have a look at the TCR repertoire and really see where the differences are. Now, as we know, of course, this kind of, let's say, differential analysis is quite difficult to do at the TCR level because TCRs are unique to each individual. So that means that we typically can't rely on having these TCRs, let's say, appear across different individuals. We typically don't have a single TCR occurring only with this specific disease. Reality is often much more complex. But what we can actually do with this kind of epitope prediction models is translate that to a higher level. Because what we can do is we can do these kind of epitope predictions and then have a look to see, okay, how many hits do I have for a specific epitope, for a specific antigen, or for a specific pathogen, and compare that between our different conditions. So for instance, here, if you compare these two groups, we see that we have on both sides, we have predictions for both the yellow epitope and the green epitope, but we can already see, let's say visually in this case, because it's simply just an illustration, that the control group has many more yellow epitope predictions. So therefore, there might be some sort of relationship between this yellow epitope and the condition that we are analyzing. So what I'm describing here is kind of a typical, say, biomarker discovery pipeline, but using this kind of epitope prediction modeling. And so the example that I'd actually like to give you here is, again, a study that we recently published from my lab based on multiple sclerosis, where we collected a lot of TCR data from different prior studies. We included, I believe, TCR data from 8 or 9 prior studies where they actually had TCR data of people with multiple sclerosis and compared them either to healthy controls or non-healthy controls. So these were people with different pathologies. They, in addition, had different types of samples, such as CFS, as well as PBMCs. And what we wanted to do was actually have a closer look at, okay, what is actually differentiating those people that actually have multiple sclerosis from those that do not. And so one of the things we did, we took all of this data, so these were several hundreds of TCR repertoires, and we ran them all through ImmuneWatch Detect. So that actually meant that for each repertoire, we actually got a list of hundreds and hundreds of epitopes that were predicted to be present in these repertoires. So this kind of just gives you an overview of the most prominent ones. So on the x-axis here are the different epitopes for which we actually got predictions. And on the y-axis were these different studies, as well as the different repertoires sizes that we actually had. So the size of the repertoires, as well as the different subtypes. So we had CD4 subtypes, CD8 subtypes, we had the mix. And as you can already kind of see, we actually got quite a widespread spurious epitope predictions across these different repertoires, which, let's say, the most prominent ones, of course, being to those that had the largest size. But of course, we were interested in, okay, what actually now differentiates multiple sclerosis patients from controls based on TCR epitope predictions. And so if we actually had, let's say, a closer look at the different, let's say, pathologies that underlie, of course, the epitope predictions, what we could see was that we actually found a clear enrichment for EBV-specific epitope annotations in our multiple sclerosis patients. As you can see, this was actually quite significant. And we did not find the same association with either CMV, so the cytomegalovirus, as well as just simply HIV, for which you could also do quite a lot of epitope predictions. So there was a clear EBV enrichment in multiple sclerosis patients. And now for some studies, we actually had a difference in CD4 and CD8 T cells because they were actually sorted out separately. And if you had a closer look in those cases, what we actually found was that this signal was primarily present in the CD8 T cells, but not in the CD4 T cells. So it's actually quite an interesting finding. And this actually relates back, of course, to the very prominent study that actually came out a few years ago, which actually linked the Epstein-Barr virus to multiple sclerosis. So there is a clear relationship between having EBV and multiple sclerosis. And this is also something that we very clearly now see when looking at the TCR repertoires by simply doing these epitope predictions at these levels. And actually seeing this enrichment of EBV-specific TCRs in these multiple sclerosis patients. And now, again, this is something that we delved much deeper into in this paper, where we actually did quite a lot of stimulation assays. And I do, again, invite you to, if you're interested in this, have a closer look at the paper for which the citation here is at the bottom. Now, this, of course, is simply, let's say, biomarker discovery. And this is still one step before what I would call diagnostics, where we are, let's say, taking this one step further, because if we know that there are specific signals in the TCR repertoire that we can associate it with a specific pathology, then comes the question, okay, can we actually then use these kind of epitope prediction models to actually do specific diagnostics? And I'm going to show you one specific case here, because this is actually quite, let's say, a topic that's quite of interest of me, because it's really, let's say, for me, the best application of epitope-specific models that really prove that they work, let's say, on a completely independent fashion. Because what you're really trying to do here is simply based on TCR sequencing, you get repertoires for, say, healthy individuals and individuals that have a specific, let's say, infectious disease in this case. And you're going to try to distinguish between those individuals that have this disease from those that are healthy, simply based on your epitope-specific predictions for epitopes known to be associated with that disease. This actually means that these epitope-specific prediction models should be used as a specific classifier for the disease, even though they were never trained as such. So what we're really doing here is going from a prediction that is actually done at the molecular level. So we're simply predicting TCRs interacting with epitopes, and we're translating that to the patient level, which is really the promise of the technology, where we can then distinguish those individuals that are healthy from those individuals that have a specific acute infection. Now, the specific case that I'm going to talk about here today is on SARS-CoV-2, so COVID-specific patients, where we actually used a data set of 80 patients that had COVID-19 and compared that to a data set of 80 individuals which were, let's say, healthy or at least SARS-CoV-2 negative. And what we did was we simply had a look at, okay, if we now predict the amount of TCRs that are SARS-CoV-2 specific, can we actually use that, let's say, as a diagnostic tool to differentiate those individuals that actually have COVID from those individuals that do not? And of course, I'm happy to report that this actually worked quite well. So we're simply taking the output of these prediction models, counting how many TCRs we get against SARS-CoV-2 epitopes, and then simply using that as the basis for our classifier. And so what we get is actually this ROC curve. So this is a receiver operating characteristic curve, again showing the trade-off between false positives and true positives, where we get an AUC of 0.8. But what actually excited me the most was the fact that we were able to get this specific dot here, which actually meant that we were able to classify 50% of the COVID patients perfectly with absolutely no false positive hits. So not a single healthy patient had more SARS-CoV-2 specific TCRs than half of our COVID patients, even though we were, of course, only limited to those epitopes in our database. So these were only 773 epitopes across 10 MHC variants, which, of course, does not cover the entire population. We also did not account for cross-reactivity. And we, of course, used simply full T-cell receptor repertoire. So we didn't do any prior sorting on, let's say, activated T cells or memory T cells. So this was just purely based on a full TCR receptor sequencing experiment. And we could already get such a powerful signal simply based on epitope prediction models. So this really shows you the potential of using these kind of prediction models to actually really delve deeper into specific diseases and how we can really go from, let's say, a molecular prediction to a patient-level prediction. Now, So these were kind of the few cases that I quickly wanted to illustrate in this webinar. I didn't have too much time to really delve deeper into each case, but I'm hoping that this kind of already gives you a bit of a perspective of what is currently already possible with these kind of epitope prediction models. Now, I'd just like to spend a few slides on this problem of the unseen epitope prediction, because this is something that everybody is, of course, working towards. And of course, this will actually unlock a lot of new applications once we actually have this unseen epitope prediction model. And a lot of the work that's already been done has really kind of illustrated that the limiting factor that is currently hindering us from having this kind of unseen epitope prediction model is really the data that is available. However, there is another problem that not a lot of people have really considered, which is what even unseen epitope prediction would look like. So I'm going back to my example that I showed you in the beginning, where we have a TCR that we find in a typical repertoire. We have, let's say, an epitope, and we simply want to have a prediction, is this a binder, yes or no? And if we have an unseen epitope prediction model, we should be able to test out any epitope that we want, and this is really the promise of the technology. However, the potential epitope space is huge. We're really talking about something that has, let's say, if you're doing this completely unbiased, you have, let's say, even with the most limited HLA class 1, 9 to the 20 to the power 9th, potential epitopes that you might want to screen. Even if you are limited to, let's say, a single pathogen or a single vaccine, you're still checking hundreds and hundreds of epitopes, and doing this kind of prediction for each will really give you probably a lot of false positive hits and will not really be what you really want to do, especially if your algorithm is quite slow. So this is actually something that we have actually been conceptually thinking of, okay, what do you actually want when you're actually doing this kind of unseen epitope prediction? And so one of the things that we've actually started implementing is not doing, let's say, a prediction based on you have a TCR and an epitope, but what you actually might want is really going from a TCR to a specific motif. So, for instance, in this case, you have this TCR, and what would come out of this algorithm would be, for instance, this specific motif. So this is actually one of the features that we've already implemented in ImmuneWatch Detect, where if you give it a TCR and you actually enable this kind of motif parameter, it will actually output a specific motif that the algorithm will actually think it will be able to bind. Now, this is actually quite an interesting example because, of course, we gave it this TCR and this motif came out. But this was actually one of the test cases that we had because this was a TCR that was withheld from our training dataset that bound an epitope that was also withheld from our training dataset. So this was actually a fully unseen epitope prediction that we were trying to do here, because we're trying to make a prediction for an epitope that is not in our training dataset. And what you can kind of see, so this was this RLR epitope that we should try to make a prediction for, is that you can see here in position 2, we have this very prominent leucine signal, which is, of course, also present in our completely unseen epitope, as well as we have this proline on position 4, which also quite nicely matches. Now, of course, this leucine here is not unsurprising, given that this is probably an HLA A0201 epitope. And in fact, this already kind of illustrates that this algorithm was actually already able to learn this kind of recognition patterns for this specific HLA molecule, which it could then use to actually do this kind of unseen epitope prediction. So it's actually quite an exciting result that we're currently pursuing more on. And we're really hoping to push this field a lot forward based on new data and new extensions to our algorithms to really try to piece apart this kind of unseen epitope prediction. But of course, this is all towards the future. But what you can currently already do with the current state of, let's say, seen epitope prediction is really delve deeper into both repertoire annotation, the response stratification, repertoire stratification, as well as the first signs of diagnostics. And I believe this was my last slide. Lastly, I'd very quickly like to take the advantage of this webinar to advertise our upcoming Antwerp TCR meeting next year in Antwerp, of course, on the 27th and 28th of May, where we have quite let's say, a prominent line of keynote speakers that will come and talk about really the state of the TCR field as it currently is. So I do hope that many of you will be able to come to this meeting, as we are actually quite proud of it.
Thank you so much, Peter. I've been having a look at the end and at the questions, and there's already quite some in. But before we go to that, real quickly, if you are now interested and want to try out ImmuneWatch Detect yourself, you can very easily do this by going to immunewatch.com/detect. We have a demo available for industry partners, but also a free license for people in academia that where you can test up to 10 samples per month. Thank you so much, Peter. I think I've seen 5 questions here, so let me start with a quick and easy one, I would say here. Maybe also on the next slide, if you want to now ask questions by live, you can also raise your hand if you want to, and I can open up your microphone and camera to ask a question live to Peter as well. But the first one being here, could I have the DOE number of the paper cited of the repertoire certification slide with Epstein-Barr virus? Because bioRxiv does not have it for me.
Yeah, indeed. So currently, I don't have a DOE yet, and that is because bioRxiv is still currently in the process of, let's say, evaluating the paper for submission. So it will actually be, I say it should be online. Let's say I was hoping now already, but bioRxiv is being a bit slow. But if you want, we can, let's say, pin it to, let's say, the recording afterwards when it does become available.
Okay, thank you. Another question. Thank you for the presentation. How do you deal with the many-to-many relationship between TCR epitopes in your machine learning prediction algorithm? Do you filter out the results by the context, like the most prominent disease in the repertoire?
That's a very good question. This is something that we've actually put a lot of thought into already. This is actually why we can actually run detect in several modes. The standard mode will actually select, let's say, the most, I want to say, prevalent epitope. So we know that some epitopes are, of course, more common. Because detect is actually based on, let's say, the information content that is contained with the TCR sequence, it will actually try to judge, okay, how certain am I that a specific epitope will bind a TCR? And it will actually select, it will actually only give a prediction if it is in the standard mode, if it is certain that a TCR can bind a specific epitope and no other one. So this is really what I mean by talking about specificity. We actually, of course, know that TCRs are more broadly specific, but as long as there's no, let's say, indications of this occurring, detect will be very specific. Okay, this is probably going to be this epitope because this is the one that I've observed in the database. So therefore, you can probably use that to kind of continue. We do also have an option of having searching for specific epitopes. So if you already know your disease of interest, you can actually already select out, okay, these are going to be my epitopes that I'm really going to be interested in. And then the algorithm will prioritize those, and you'll get those in your output. But really the fact that, of course, TCR epitopes are many-to-many, as you quite clearly state, is why we are migrating more towards this like motive level predictions. Because this is really where I see the kind of the future of TCR epitope prediction, where we're not predicting a single epitope for a single TCR, but what you actually get is a specific motive, because it's really what the TCR is looking at. It's looking at specific patterns in the epitope. And this is what the algorithm should predict. And then you can use that motive to kind of already go and screen any kind of disease or antigen of interest to see if you actually find any matches within the protein sequences. So this is why we're also offering this kind of motive level prediction, because we really think that this will be quite useful, let's say, in this kind of context, especially as we get more epitope data.
Thank you, Peter. Again, I want to stress, if you want to ask a question live, feel free to raise your hand with the feature on the bottom of your Google Drive, your Google Meet link here. The next question comes from Sergio. One of the issues is the pairing of alpha and beta, which is right now not possible with bulk RNA sequencing, and single-cell RNA sequencing being very expensive. Is beta sequencing only sufficient for decent predictions?
Yeah, this is again a very good question. Now, we know that paired sequencing is better for the predictions. We've published quite a few studies on this. In fact, this was actually one of the most prominent findings of, let's say, the IMREP benchmark in 2022, so not the one that I showed today, where we know that pair chain predictions will always be better than, let's say, single chain predictions, which makes sense because then you have the full TCR. Now, that being said, a lot of actually the results that I actually showed you today were on bulk TCR sequencing. So these were actually unpaired predictions. So everything from, let's say, the diagnostic classifier to a lot of the multiple sclerosis stuff were simply done based on single chain, often beta chain predictions. And one of the big advantages that we have, let's say, with kind of the detect algorithm, and this is why we also build it that way, is that it will really try to judge, do I have enough information to actually make my prediction? So we can actually apply the same algorithm, and actually the detect algorithm is actually quite unique in that, on both bulk TCR sequencing as well as single cell sequencing data. Because if you simply give it unpaired sequencing, what it will actually do is it will actually judge, okay, do I have enough information in my beta chain TCR to make a prediction for a specific epitope? Or do I actually need more information? Do I actually need the alpha chain as well? And therefore, it will give maybe a prediction, but at a much lower score. So well, ideally, you do need single cell data to get really the best kind of predictions. What we've seen, let's say, in our own studies, is that very often unpaired TCR sequences will already give you quite a lot of signal.
Okay, thank you, Pieter. I saw a hand by Jürgen, but it's gone by now. Is it? Okay, yes. I'll allow you on the floor, Jürgen. Yes, you should have permission to open your mic and camera. Is it working out, Jürgen? On the bottom, you can unmute or open up your mic, camera. In the meantime, I can maybe ask another question. That's a sad face. In the meantime, I maybe can ask another question that's already asked in the chat as well for Jürgen to figure out to open his microphone. Sergio also asks, no, this one I already, yeah, okay, no, another question. For the AML patients, you both did DNA and RNA TCR sequencing. Could you comment on the need for both DNA and RNA?
So both DNA and RNA have their own unique advantages. DNA is typically a bit more quantitative because, of course, you only have one DNA molecule per T cell. While RNA, you have multiple RNAs per cell, so that can actually be a bit more sensitive towards the presence of specific TCRs in your sample. So we've actually done, let's say, in our own labs, a mix of both, depending on the exact need. We know that if we, let's say, have a low quantity of cells, we typically go for RNA, while, let's say, if we really want a quantitative assignment of exactly how many T cells are present in a specific sample, then we might opt for DNA. Now, if you have the opportunity, of course, to do both, you can kind of, let's say, of course, mix the best of both worlds.
Okay. Thank you, Pieter. Can we give it another shot, Jürgen? Can you try and unmute yourself? Oh, sorry about that. Maybe refreshing the browser could work. Maybe it's about the permissions with the microphone. I don't know. I'm sorry about that. You can always leave a comment in the questions as well if you want to type it out. Sorry about that. Maybe two more questions here in the chat before we wrap up. Jeroen asks, for predicting unseen epitopes, wouldn't it be a good idea to model TCRs in 3D and use that as a starting point?
Yes, that's actually a very good comment, Jürgen. I know that there are actually quite a few labs that are actually exploring, let's say, this approach towards unseen epitope predictions, where they're really taking the TCR structure, doing predictions, and then kind of overlaying, for instance, the specific epitopes, or at least the PMHC complex. Now, the big disadvantage, of course, there is that we don't have a lot of good 3D structures for TCR complexes. I think last time I checked in PDB, we have maybe 100 or 200, and most of them don't even have the epitope complex. So this is one of the, let's say, biggest challenges still, is that we don't have a lot of data, say, on both the 3D molecular side, as well as even on the epitope-specific TCR side. You also have to kind of consider that, of course, if you have large repertoires, doing this kind of 3D modeling will become computationally quite intensive. So I do expect that we're going to, even in the future, going to need a mix of both kinds of models. But so far, let's say my own research has focused more on the sequence aspects, simply because we have a bit more data there. And it actually allows us to do kind of this kind of repertoire annotations at quite a speedy pace.
Okay, we managed to get Jürgen's question in the chat here. So the last two questions and then we wrap up. How well does the prediction work for gamma delta TCRs?
Yeah, that's actually a very good question again. And I'm actually quite excited about the fact that in actually the Immunology Tech database, we actually have gamma delta TCR data. So I don't think there's a lot of, I don't think there's any gamma delta TCR even in the public repositories. I might be wrong. But we can actually do predictions for gamma delta TCRs. Now, unfortunately, there hasn't been that much work done on gamma delta TCRs in the field. The field is much more focused on the alpha beta TCRs, which I find a bit unfortunate. But we do have quite a lot of gamma delta TCR data in there, which actually does kind of show the diversity of gamma delta TCR targets as well, where we can actually do predictions both for HLA dependent as well as HLA independent antigens.
All right, super. Thanks for that, Jürgen. Sorry about the technical problem. Last one. Here I'm opening the questions again. So when combining different TCR-seq data sets, do you need to correct for differences induced by library preparation kits, assuming that different kits are used, actually?
Yeah, that's an extremely good question. So this is actually something that we observe. If you have different TCR sequencing kits, you will actually see slight changes in the repertoires that come out. What you see quite prominently is that you'll see different usages of V and J genes that cannot be, let's say, biologically explained. And this is likely to do with, of course, the specific primers that were used in the sequencing kits. So you should actually be aware of this specific issue, that if you have a repertoire that was done with a specific sequencing kit, you can't just simply compare that to a repertoire that was done with a different sequencing kit, because you'll always find a signal that will be kit dependent. So that is actually why in a lot of our studies, we really make sure that if we're comparing two samples, that they should be done using the same sequencing kit. Now, of course, for the specific multiple sclerosis case that I actually explained here, we actually had to combine data from a lot of different studies, and they actually all used quite a lot of different sequencing kits. And we actually go a lot into detail into our paper there on, first of all, the differences that we observe, which are actually quite prominent and quite interesting. And secondly, actually an extensive correction, how we can actually correct for the presence of these sequencing kits in such a way that we could actually were able to compare between these different studies quite well. And it actually worked quite good, and we were able to validate that as well.
Okay. Thank you, Pieter. We're going to wrap up here. We're 5 minutes over time. There's 2 more questions in the chat. Feel free to reach out to Pieter directly. I'm sure he'll answer your questions. Thank you so much for attending, and we hope to see you in the future as well. Goodbye.